introduction. And ladies and gentlemen, it's my honor to stand here and give my presentation. And my topic is understanding the floral shape variation in Sinningia. And in my project, I will going to use the three-dimensional geometric multimetric approach to reveal more detail about the flower. Especially, I want to use this strategy to answer some questions in biology, especially in the development of biology. I think I will start from the how special this Sinningia is all about the flower. So in previous study, some botanists already know that Sinningia have various floral morphology, and this morphology variation is defined as the Polynesian Syndrome. Here comes some morphology like the floral shape and floral color. So you can see here, every block means the different type of pollinator. Like the red flower is maybe pollinated by the hummingbird, and the purple flower or yellow flower is pollinated by honeybee. So let's look more detail about the flower. So here's the process of their pollination. You can see that the left top side is the hummingbird pollinated flower and the hummingbird, and the top right side is the bumblebee, and left hand side is the bat pollinated flower. So how's the process? You can see here is the dissected view of the pollinator and the flower. In this tree diagram, it's showing the bumblebee pollinated flower. So you can see that here we have three different. So in this tree diagram, you can see this is the three different species, and these three different species have specific pollinator to help them to pollinate. So you can see the body size is different, and also correspond to the different floral shape. So here comes the short conclusion is that the floral shape maybe brings the physically restrict the visiting of the pollinator. So in the process of evolutionary biology, maybe we call it is the co-evolution between the flower and the pollinator. So we say the evolution is triggered by the pollinator. So for the botanist or the biologist, the challenge is we want to know about the relationship between the phenotype and somehow like the gene. So in the very beginning, the challenge is to define this trait, to quantify this trait. So in here, we can see traditionally biologists can describe the shape, use several terms, which is a discrete term. It's like in the different flower shape, you can use different adjective term to describe that. But how about this? If we have a group of species, they share the similar floral shape, but with little differences. So how can we still use this strategy to describe the shape differences? So here comes the idea is how can we whether we can precisely and accurately to quantify the shape variation. So the 3D maybe provide a good solution to achieve this goal. And further information I want to bring here is about the evolution background of this species. In Sinningia, we can see here's the tree, evolutionary tree, and here is the side view of the floral diagram. And I put some asterisk star here. The red is the hummingbird pollinated, and the blue star is bumblebee pollinated. You can see the development or the evolution is independent. So you can see they are nested. So it comes that how floral shape comes to this morphology. Maybe they share the same mechanism in the evolution. So here make me think about generate a research to answer this question, how the floral shape associate with the pollinator type. So let's look at the detail of the flower. On the left-hand side is bee-pollinated flower. On the right-hand side is hummingbird-pollinated flower. You can see they really shows the very big differences between each other. So, following, I will go to my material method. So the material, actually in Sinningia genus, we have more than 80 species, but up to now, I only sample 13 species for current study. So among 13 species, 4 is bee-pollinated and 9 is hummingbird-pollinated. This ratio is coincident with the 80 species. Then for each species, the specimen will prepare in fresh or either in ethanol-fixed sample. And the strategy to capture the three-dimensional information, I also use the micro-CT. Then how to analyze the shape, I use the landmark-based three-dimensional geometric morphometric analysis. And here's the workflow. The first step, the specimen preparation, is the gardening work. So until they're flowering, and I send them to the micro-CT and put on the stage and do the sectioning scanning. Then the result will come like this. It's a slide sectioning image. So in each slice, we have a base and our object is here. So we implement the image processing on this slide and then reconstruct the three-dimensional image to become a volumetric image and even more the surface image. Then we identify the landmark on this image. Then the following is the GM analysis work. So for the detail, each step. The first of all is the capture of the three-dimensional image. So in the very beginning is the volumetric image. And in the volumetric image, we may have some noise in the background. So the process is like this. The scanning, the result is a sliding image. So, and then it is the volumetric image. Some sparkle here, it is the noise. So furthermore, we improve the image quality. So there, totally we have 5 steps. For the first one, remove the base. And then the second and the third is all about a series of image processing. And anyway, we get improved image here. Then we assemble, reconstruct the image like this. This is the volumetric image without the noise here. By the way, we build this system in the MATLAB. And then finally, we convert the volumetric image into the surface image like this. And then we use this surface image to identify the landmark. So here I want to give some definition of our landmark. Totally, we use the 420 landmark to describe each flower specimen. So for a certain species, 3 specimens of each species, we have 420 landmark for each specimen. And the detail is like this. Every flower has 5 lobes. And each petal or each lobe, we have the lobe edge, and the lobe boundary, and lobe mirror, and tube boundary, and the tube mirror. So totally, we will have 420 landmark. And the process is like this. In the MATLAB, we develop a GUI system. Then we manually identify the contour and the mirror and the boundary. Then we just equally separate the distance along the contour, so we can get the landmark distribution like this. So it is our input data for the GM analysis. So how is the GM? GM includes 2 steps. The first step is generalized procrusted analysis. It is used to retain the information about the shape and remove others. And the second step is the PCA, is to summarize the shape variation. So the concept of the GPA, the generalized procrusted analysis, is like this. The first step, it is the initial configuration. It's a specimen. I use the 2D image as an example. So maybe we have the different location, different size, and different angle on each specimen. So the first step is translation. we move each specimen to the same gravity center, so superimpulsive. Then we have a rotation. We make each lobes in the same direction, and we scaling to make the size in the same size. So this we call the shape coordinate. Then the shape coordinate, we send that into the PCA to summarize the shape variation. So here comes the preliminary result about the shape variation. In summary, we have more than 85% of total variance can explained by first-rate PC. So the script plot is like this. Then the distribution of first-rate PC, you can see the right to plot. The x-axis is PC1, and this one is PC2, and this is the PC3. You can see that the orange triangle is hummingbird pollinated flower, and the blue circle is bee pollinated flower. You can see in the PC1 and PC2 scatter plot that can simply separate into the two groups. And following in the PC3 and PC2 and PC3 script plot, maybe they have more complicated variation in this axis. So let's revisualize the variation in the specimens. In PC1, we can see here is the mean shape, and two direction is the positive extreme and negative extreme of the shape variation. You can see that in the different extreme shows the very different floral shape. And in PC2 and PC3 shows similar. The result, the positive and negative extreme is very different. And simply we can literally to define the shape variation by some physical trait. It's like in PC1 we have the tube curvature, more bending downward, bending upward. Or in PC2 we have the ventral tube protruding, like here in the very big bottom. And PC3 and so on. Then how this distribution bring us some any idea from this result can give us some new idea. So here comes some current discussion here. In the PC1 and PC3 scatter plot, we can see that actually this flower doesn't exist in our sample data, but we look forward on the different other genus or other species. We can see that this floral shape exists in some other species, and not only this case, but also this in the PC2 positive extreme. We can see this shape. So which means that in the PC1, PC2, PC3 analysis, it constructs a shape cloud or the shape space, which means maybe the floral development is all distributing on this space. And this space maybe can bring us provide us some strategy in the conservation biology to help us to know that maybe there are some unidentified species in the wild that is worth to further to work on. So in the short summary, in current study, 3D approach was successfully applied to investigate the floral shape and to know the relationship between the bee-pollinated and hummingbird-pollinated flower, and GM is successfully to revealing the shape variation. And combining the 3D and GM enable to study the complex phenotype. And for future work, as I mentioned previously, I only sample 13 species. Maybe it is not enough to say to have a complete result, but in here I give a demonstration to utilize this strategy. Then maybe I can use this system to investigate the developmental modularity for the floral shape to answer some question in the biology, in the developmental biology. And then I want to thank several persons, especially the advisor and some co-worker in the lab, and especially the technician help us to get the CT data. And thanks for your attention. Thank you.
Thank you very much. Any questions or comments? said you need more samples, but I think very successful.
Yeah.
What happens when you're thinking about another samples?
So you mean why I'm going to get more samples, yes, right? Yes. Because let me show more details about the Sinningia. Here's their biodistribution, species distribution, and here's the tree. Actually, I only select the representative flower shape in current study, so I think it is not enough for me to answer the question on the developmental biology. So that's why I want to get more specimen.
Any questions, comments? Yes, thank you very much for successful result and the presentation.